along with myself, there is Kerry McLeod here. She's our Head of Supporter Relations, and Juliet Tweed, our Marketing and Engagement Coordinator. It's really wonderful to finally be back doing in-person events after a couple of years of cancellations due to COVID. I'd also like to welcome Dr Melanie McConnell here, who is going to be speaking to us this evening. But before she gets started, I'll just give you a very brief introduction to the Neurological Foundation, if I can find my clicker. So the Neurological Foundation was established 51 years ago when a group of neurologists realised the importance of establishing an organisation to fund research into neurological diseases and disorders. 1 in 5 New Zealanders are affected by a neurological condition, and there are over 700 neurological conditions. The Foundation receives absolutely no government funding, so we're completely reliant on the generosity of New Zealanders supporting us. In the last year, the Foundation has funded close to $5 million worth of neurological research. Because of the generosity of our supporters, that's nearly $1 million more than in the previous year. So, as well as funding research, the Foundation has also made a commitment of funding the Neurological Foundation Human Brain Bank for another 5 years. This resource is one of only 8 in the world. It's a world-class resource, and it's headed up by Professor Sir Richard Fall. The Foundation has also committed to funding the Chair of Clinical Neurology for another 5 years. The focus of this since 2008 has been on stroke and the clot retrieval procedure. The Chair of Clinical Neurology is headed up by Professor Alan Barber. At our most recent funding round at the end of June, $2.2 million worth of neurological research is being funded, and our next funding round is in November this year, so we have 2 funding rounds every year. We are so incredibly grateful for every single dollar that we receive in donations. Neurological research cannot continue in New Zealand without the support of our donors, so thank you so much. It now gives me great pleasure to introduce you to tonight's speaker, Dr Melanie McConnell. The Foundation has funded Melanie and her research a number of times over the last 10 years. She's a senior lecturer at the Genetics School of Biological Sciences at Victoria University here in Wellington. Melanie will be speaking this evening about her latest research, which is looking into why brain cancers resist therapy. So there will be an opportunity after Melanie's talk to ask questions. So after her talk, we will come around with a microphone that you can ask questions through. So thank you, Melanie.
So, I'll probably take this off. I wear my mask all day, every day, and I kind of forget that I'm wearing it, but it does make it a little bit hard for you to hear me. So you look like a kind of a low-risk crowd, I think. I work in a university with a whole lot of 20-year-old students, and they are not low risk. Okay, so really happy to be here to talk to you a little bit about our research. I would start with some introductions. Ko Melanie McConnell, toka whenua, kei rangaho mati pukupuku. I'm a cancer researcher, and that's really how I define myself. And I'm really interested, and I have been for a while now, particularly about brain cancer. So what I'm going to do tonight is sort of take you through my story a little bit, how I got to where I am, and how where we're going to be going. Okay, so I grew up on the Kapiti coast. So I went to Paraparaumu College. There's probably no alumni from Paraparaumu College here, but I grew up in Kapiti. And this is, and if I stand on this side, we'll still get the mic. So Kapiti Island here, this is what I would call my raraho. This is my mountain of of significance. And this is actually a stock picture of the river where I grew up. So I grew up in Otahanga, and if you Google New Zealand River, you get this picture. So it was a very sort of idyllic suburban childhood. A little bit of background, I whakapapa to a couple of significant vessels. One is the Whitby. I'm a direct descendant of the very first colonist to ever die in the colony of Nelson. So my grandfather's great-grandfather is buried here on Hawlashore Island. He came out, he thought he'd bought a farm from the New Zealand Company, and he got to Wellington and discovered maybe he didn't have a farm, and then he picked up typhoid and he was dead by the time he got to Nelson. So his wife arrived on the next ship with their 4 small children, including my grandfather's grandfather. And I also whakapapatu to Arawa, and according to the family legends, we are distantly related to Te Mura Morrison. So if you take careful pathways, we can find a connection to Te Mura Morrison. So it's just a little bit about where I come from. I've got 2 teenage daughters, I have a husband, I have a veggie garden which is really sadly neglected at the moment. It's too wet to be out gardening. I'm a 3-season gardener. And I describe myself, so I'm a pukinga matua at the Victoria University of Wellington, and cancer researcher. Okay, I'm not allowed to show pictures of my family anymore. They don't want me showing their pictures, so this is my dog. She works very hard. Here she was at a street appeal for the Cancer Society. All right, so I started my research journey with this organism. Now, do we have any sheep farmers in the room? Anyone come from a sheep farming background? No. So you won't recognise this disease. This disease is called scabby mouth, and it's caused by a pox virus. So I got my start in research in virology. And it's a pox virus, and it is a really stable virus. These scabs fall off the sheep, and they can survive in the grass for like 10 years or more. So it's a very sort of therapy-resistant kind of problem. And if you're exposed to this, you can get these blisters on your fingers. And so in my first research project, I managed to stab myself with a syringe full of this virus that I had just isolated. And at that stage, I decided I needed to leave virology because it wasn't a safe place for me to be. And I made the move to cancer research. So my PhD was in this tumour called Wilms tumour, and this is a kidney cancer, and it's a paediatric disease. So this is a kidney, and all of this here is tumour. This is a boy called William who developed a Wilms tumour, and his family have set up a foundation to try and support people with this cancer. It's a fairly rare cancer. It's kind of a nasty one. There's no good cancers. But this is a kind of a nasty cancer. And the reason I'm telling you this background is that it's really important to how I do my research, basically. Okay, and then after my PhD, I moved to New York City, and we lived in New York for about 10 years. And this is New York. And here I worked in leukemia, so it's another kind of cancer. It's another tissue of the body. And I worked really closely here with scientists and with doctors, and we worked on trying to understand how leukemia cells tick. What makes them tick? What keeps them alive? How do we kill them? And this is just a picture of some. You probably can't tell. But do we have any pathologists in the room? Okay, I'm safe. These are some really abnormal looking blood cells. So I've had this quite broad background, and I'm living overseas, I've got a young child, decided it was time to come home. And an opportunity arose, and that opportunity is very much how many researchers, how a research career goes. You rely on serendipity. And so serendipitously, at the time that I was looking to come back to New Zealand, a man called Sir Roy Mackenzie wanted to fund some research into stem cells. I believe this was competitive philanthropy because Peter Jackson had just funded some people in Los Angeles to work on stem cells, and that annoyed Sir Roy, and he thought that we should be working on stem cells here in New Zealand. So he fronted up with some money for a fellowship, and it was kind of a weird confluence of events. He wanted people to work on stem cells, and I had been working on these leukemia cells, which are very much like stem cells. And so there was Sir Roy wanting to support this work, and then me, and then there was a neurosurgeon. And that neurosurgeon's name was Martin Hun. Mr. Hun's he's a fabulous surgeon. He's in Melbourne now. I think he wants to come back, but yeah, hopefully he comes back. So I ended up, through a kind of a weird convergence of factors, saying, yes, I can work on brain cancer. We know that there are stem cell-like cells in brain cancer, and how different can it be to leukemia or to kidney cancer or to viruses? Honestly, I can do that. So they believed me, and they gave me the fellowship, which was amazing, and I had a baby in the middle of it, and that extended it out for 3 years rather than 2 years. And so I was working at this place called the Mulligan Institute. Now, if any of you have been in Wellington, this sits on the very edge of the hill of the university campus, and it's a round building. And this is a focused research institute. It's all about research. Teaching is considered a distraction. Having a family is considered a distraction. It's really about research. And so these guys were really instrumental. And so working with the team here, we set up the first dedicated brain cancer research program. And then I started learning, and I learned a lot about brain cancer. And the thing that struck me the most was what I learned is the brain is a really unique organ. You know, the brain is an incredible organ. We don't understand anything really about how it works. So if you think about a kidney, it's quite simple. It's a filter. It's a filter and it's a concentrator. If we think about our blood cells, they're there as immune cells. They're out there. They protect us against things. It's really very simple. And then I got to the brain. And the brain's a really, really unusual organ. We know it's really important. We know it uses 25% of the energy that we take in. We know there's electrical signals. We know there's different areas of the brain, but we really don't understand how it ticks. And so it was a really, really interesting challenge to understand how an organ that's so important and so difficult to understand, like how are we going to unpick what's going on in a disease setting? So we recruited, starting in about 2006, we recruited a bunch of tumour samples. 2006, 2007, 2008, we collected a lot of tumour samples from people who had glioblastomas who were having surgery. We got them into the lab and we started trying to dissect them. What is going on in these cells? Why are they so unusual? And then I ended up taking a teaching position at Victoria University. And so I moved 50 metres from the Mulligan into the next building. And I now teach undergraduate biology, and I try to inspire the students coming through to love unusual, naughty, wicked cells the way that I do. So I've still been working on the brain cancer research, and now I've got a whole group of students that love this disease as much as I do. I mean, it's a horrible disease. I shouldn't say I love it, but it's an area of research that I think is really important. Okay, so what is brain cancer? That's a really hard question to answer. We say that there's about 200 different types of cancers, if you think about cancer generally. There's about 130 different types of brain cancers. So it's an incredibly diverse disease. And this is a quote from the textbook that I use to teach scientists. And it says that a cancer cell is a malignant cell. So that cancer cells break the most basic rules of cell behaviour. And they exploit every opportunity to do so. And I think that's a really accurate depiction of what cancer cells do. They don't follow any of the rules. bodies work because cells do as they're told. And when they break those rules, they stop working. And so if we think about the brain cancers, we've got benign tumours and we have malignant tumours. So a benign tumour is one that's slow growing. It will respond to treatment. Generally, it will respond to treatment. It's in a site that you can access. A malignant tumour is a very different kettle of fish. The malignant tumours have broken all the rules, and it's very hard to predict what they're going to do because they don't follow any of the rules. Also, when we talk about brain cancers, we can have the ones that have originated in the brain, or we can have the ones that have metastasized to the brain from somewhere else. So brain cancer is a really complex sort of set of conditions. So we're going to have a little bit of an anatomy lesson. One of the reasons there's 130 different types of brain cancer is that there's lots of different regions of the brain. So this is a schematic of the brain. If I walk away from the microphone, you can't hear me. And if I turn around so I can see, okay. So we've got this region up here, which is the cerebral cortex. This is the kind of the bulky bit of your brain. We've got this region in here in green, which is the cerebellum. There's the brain stem here. It's got an area called the pons in it. The spinal cord comes down here. There's a little area in here called the hypothalamus or the thalamus. The hippocampus is this area here. This is where your memories are made. All your memories are made and stored in the hippocampus. And every part of your brain has a different function. Oh, I've forgotten one part of the brain. So here's the very edge of the brain. And the other thing, because the brain is such a precious organ, it's protected in a way that lots of our other organs aren't. So as well as, I've got a back button, so as well as the brain itself, we have the meninges, or the layers that surround the brain. So shown here is the very edge of the brain, and we know that the brain is crinkled and wrinkled. This is one of those crinkles. Find the right button. This is one of our crinkles in the brain, and we have these layers here, which are the meninges, and then we have the cranium, or the bone, which surrounds it, and the bone provides a really sturdy protection for the brain. Stops things from the outside coming in. If you have a tumour on the inside of this brain, there's nowhere for it to go, and so you get a lot of pressure building up. And then the other thing we have on top of, well, some of us have on top of our brain is skin, and in that skin sometimes there's even some hair. Okay, so we've got our brain. It's got all these different regions. It's got the meninges around it, and then the brain. Now, all of those different areas can potentially become cancerous. They all have different layers. So we've got the arachnoid layer, and then we have the subarachnoid layer. So if someone has a subarachnoid hemorrhage, for example, it's a hemorrhage in this area under these meninges. Okay. And then, as I said, every region of the brain has a different function. So this is an indication of those different functions of the brain. So smell, and is it right at the front of the brain? Sight, vision is right at the back of the brain. Touch and pressure are at the top. Concentration, planning, problem solving is up here near the speech region. Facial recognition is right down here near the base. The cerebellum here controls your coordination, your motor skills. The brain stem down here, this region that includes the pons, this is your basic functions: heartbeat, breathing, swallowing. So, depending on where the tumour arises, you get different symptoms. There are different things that can happen. If you get a tumour in this region here, the effect of that might be a speech problem. You get something back here, you might have hallucinations. It's a fascinating organ. It really is. Okay, so I am not an anatomist. My focus, my interest is on the individual cells that make up the tumour. So there's all this big scale stuff. It's all very interesting. But what really gets me excited is looking at the individual cells and all the 100 billion of them that make up the brain, like how they all work. So we have, and this is sort of a schematic of the kinds of cells we have. So we have the neurons. The neurons of these cells here shown in grey, and they're the business cells. They're the ones that have the electrical signals, and they make up, have I animated this? No, the animation's not working, that's okay. So the neurons make up about 10 to 50% roughly of the cells in the brain, depending on which part of the brain. And they're the ones that transmit the signals, they're the ones that make our memories, they're the ones that allow the brain to function. And so for a long time, people cared about the neurons and they ignored everything else in the brain. And there are still some people who do that. We have these cells here called oligodendrocytes, and these are the ones that act as the insulation layer for the nerves. Nerves transmit electrical signals. They need to have insulation around them. And so there are cells that make the insulation. So these are the ones that make the myelin sheath. And if you're going to come to the talk tomorrow, my colleague Anne Laflamme is going to talk about demyelination, which is the loss of the insulation around the nerves. Now, this is my favourite cell here. This is the astrocyte. So an astrocyte or a glial cell, they were kind of ignored for a long time, and I think that's why I feel so, that's why I feel for them. They were ignored for a long time, but they have a really, really important role. They make up a lot of the bulk of the brain, and they do a couple of things. They take the food from the blood supply, and they give it to the neurons. So they feed the neurons, and then they take the waste away. So you can imagine what your brain would be like if your neurons weren't being fed and weren't having the rubbish taken away. So I am team astrocyte. I think these are the most important cells. So the astrocytes get the nutrients from the blood vessels. And so we have the cells that make up the blood vessels, and these cells are surrounded by what we call a pericyte. And this is this blood-brain barrier. Has anyone heard about the blood-brain barrier? So the blood-brain barrier is a set of cells that grow around the blood vessels, and they control what gets out of the blood and into the brain. Now, that's a really important thing to do. You don't want toxins, you don't want anything really getting through here that's not going to be good for the brain. So this blood-brain barrier plays a really important role in protecting brain health. And then lastly, we have these immune cells. These cells are called microglia, and they're the immune cells, and they're the ones that will hang out in the brain, and they'll wander around a little bit, and they'll look for signs of damage or signs of infection, and they will clear it up, hopefully without causing too much of an inflammation in your brain, because you can imagine if your brain's got lots of activity happening, if you've got a lot of inflammation, if you think about when you get an infection and you get like a sort of like a little pussy hangnail or something, if you've got that going on inside your brain, that's not going to go down very well. So these cells really try to keep all of that other action out of the way. So any one of these cells can acquire a mutation which turns it cancerous. And it's just kind of bad luck, really. If something happens to one of these cells, and it can just be a mistake in the way the cell is behaving, it might be an environmental exposure, it might just be bad luck. That if one of these cells gets a mutation in its DNA that changes the way that it acts, then that has the potential to become a cancer. And so that's the reason we have so many different types of cancers. We have different types of cells, we have different tissues. What's going on? I did have some things animated. Right, so here is, I've drawn a beautiful picture here. Here was my little brain, and you can see I've very carefully drawn these meninges around it. So if we think about the really common kinds of brain cancers, we can have meningioma. So about a third of all brain cancers are meningiomas. You might know someone who's had a meningioma. Anyone know anyone with a meningioma? No? So that's where the cells that make up this layer become abnormal. Most meningiomas can be accessed. You open a hole in the skull and you can cut it out. And they tend, about 85% of those tend to be responsive to treatment, and you can remove them. So we have a bunch of different, so that's the cells of the meninges. We might have cells all through the rest of the brain here that are the glial cells. And so we've got our astrocytes, my favourite cell, the astrocytes. If something goes wrong with them, we call that astrocytoma. The oligodendrocytes, those cells that make the myelin sheath, if something goes wrong with those, it's called an oligodendroglioma. This is a paediatric cancer, diffuse intrapontine glioma. These are the cells, the astrocytes, right down here in the brainstem that become mutated. So you'll see that there's a bit of a theme going on here in how these are named, yeah? Everything's got an oma at the end of it. So, neuromas, so the neural cells can be mutated, and we call those neuromas. Acoustic neuromas are mutations of the mutation in the nerves that feed the ear. Does anyone know anyone that's had an acoustic neuroma? These are, you go deaf basically because the nerve that's feeding the ear becomes so abnormal it stops working. So any of these cells can have a mutation in it. It's just bad luck, and it happens reasonably frequently. Oh, it's just a picture of the cells for you. Don't forget the astrocyte. All right. So, people think of brain cancers as being rare, and I don't know why they think of that. I know the most common cancers are colorectal cancer and breast and prostate and those kinds of things, but these are not rare tumours, and I'm really frustrated that people tell me that I work in why do I work in such a rare tumour type? So in kids, the most common kind of cancer after leukemia is brain cancer. And some of it, so there's some names up here: medulloblastoma, neuroblastoma, ependyoma, diffuse intrapontine gliomas. None of these are particularly rare tumours in kids. They hit different parts, different regions in the brain, and it's quite frequent that these happen. I don't know whether anyone knew or remembers the story of Jemima Gaisley. She had a diffuse intrapontine glioma. It's a reasonably rare kind of tumour to have, but brain tumours are really common in kids. In adults, they get less frequent. So something about getting through to adulthood protects your brain a little bit, but they still happen, and it's about 5 to 6 people out of every 100,000 will come down with a brain cancer, so the malignant form. So it's not particularly rare. They are slightly more common in men. Now, we don't understand why this is. We know that there's differences in the way the cancer cells behave between men and women. That's fascinating. We've just started trying to figure out why that is. Some cancers are more common in women than in men. The meningiomas happen much more frequently in women. The glioblastomas happen more often in men. The numbers balance out, and basically men are slightly more likely to get a brain cancer. And what's really curious is the incidence is the same in all ethnic groups. We don't have particular groups that are more or less prone to them. And the incidence hasn't really changed since the 1970s. So that tells me that whatever is causing these tumours is not something in the environment that's increasing over time. So we haven't seen an increase associated with cell phone use, for example. To me, that says that whatever is driving this is something that happens inside those cells, and it's something that's happening at about the same time over the last 50 years. So it's something that's just bad luck. It's a thing that happens, and it's not like you could have prevented it in some way. I thought we'd just have a look at some of the other cancer rates. So lung cancer occurs in about 30 people every 100,000, and so that's about 5 times more common than a brain cancer. Melanoma, 6 times more common. Colorectal cancer, can I do the maths? 7 times more common. Breast and prostate cancer, really, really common. So if we look at that incidence rate, the brain cancers, maybe they're rare. They don't happen as often as these other ones. But if we compare the incidence, so with this low incidence, what can we do about them? So we know that for breast cancer and prostate cancer and colorectal and melanoma, for all of these really common cancers, there are things that we can do to protect ourselves. We can wear sunscreen, we can do screening, we can, feel for lumps, we can do blood tests. Those kinds of things for these really common cancers are something, proactive steps that we can take. And if we look at our brain cancers, we don't have those kinds of tools in this disease. Now, the symptoms of a brain cancer depend a lot on the area of the brain and the cells that are involved, but they're actually depressingly vague. Headache, nausea, dizziness, numbness or weakness, personality change. This is not something where you can go, oh, damn it, I think I've got a brain cancer, I've got this headache. Yeah, that's exactly. So the symptoms are really hard to pick up. We don't have any screening process for brain cancers. We have no way of early detection. You can't feel a lump in your brain because your skull is protecting your brain. And because we have no blood tests that you can kind of keep an eye on. Look with your PSA and your prostate, you can keep an eye on that with the blood test. We don't have anything like that yet for brain cancer. And because we don't know what causes them, and we think the cause is just sort of intrinsically mistakes that happen in cells, we really can't prevent them. So where there's a lot of focus at the moment in the media and in the political areas about prevention and screening and diet and all these really good things, none of that's going to do anything for our incidence of brain cancer. So, I said earlier we have the benign ones and the malignant ones, and luckily most of our brain cancers are benign. They are fairly slow growing, they're in an area where your surgeon can find them, they can pull them out, and they tend to be killed by things like radiation therapy. Our malignant cancers, not so much. They're very rapidly growing and they will invade into the cell, into the tissue around them. Because they kind of move out from where they've started, your surgeons they can't get them all out. I actually asked Mr. Hun when I first started, could he please get me a couple of centimetres of normal brain tissue around the edge of the tumour so that I could have some normal tissue to have as a control? And he looked at me and he said, no, I'm a very good surgeon. But what that means is that the surgery can't get all the material out. They're protected from chemotherapy by that blood-brain barrier. And so, your body tries really hard to protect your brain from toxic chemicals. And so if you want to give someone chemotherapy, you've got to get it through that blood-brain barrier. And it adds to the problems. And then for some reason, and this is particularly the glioblastomas, and this is particularly my area of interest, they don't care if you give them radiation therapy. They just kind of shrug it off. So with all of this in mind, if your diagnosis is a malignant brain cancer, you have a cancer that's rapidly growing, it can't be removed, it's protected from chemo and it's resistant to radiation. And the prognosis generally is described as dismal in the literature. That's the official term for it. It is dismal. The death rates haven't changed since the 1960s, and this is the really depressing part. We've been doing research in glioblastoma as a worldwide scientific community since the 1960s, and we haven't actually changed the death rates. It's got a much higher rate of death than melanoma. So 4 out of 5 people that have got a malignant cancer will end up dying from their cancer. So this is higher than melanoma. Even though melanoma is 6 times more common, you're much, much more likely to die from your brain cancer. So this sounds awful. This really sounds, it's a horrible, miserable, revolting disease. And so this is actually a patient who is here in Wellington. They contributed their tumour to our first study. And so this is an MRI, and we can see we've got the skull around here, the meninges. And this gap here, this is where this person's tumour was resected. So they had their surgery. We took out, meaning Martin, not me, I'm not the surgeon. Martin took out their tumour, and they had 6 weeks of radiation therapy, and they got 6 months of chemotherapy, and while they were being bombarded with these chemicals, their tumour just kept on growing. So all of this here is tumour, and you can see that it's squashed the brain. There's lots of pressure going on here. And so this gold standard is the best that you can get anywhere in the world. Surgery, you remove as much as possible, you get some radiation, and you get chemotherapy. And sometimes it might be 6 months, or maybe you have a little bit longer. And the end result is we haven't really done anything. And this is an absolute heart-sinking moment for an oncologist or for a surgeon. But for a biologist, I'm going, oh, that's quite interesting. How did that happen? Because this goes against all the rules we know about how cancers work. They should be dying. They should not be growing and expanding and moving out. And so, from a biological perspective, this is absolutely fascinating. Okay. And there's some more really dismal statistics. The median survival, so the average that you would survive after diagnosis is 15 months. Lots of people die within the first few months. Your chance of surviving out to 5 years is about 7%. But people do survive that long time. There are people that make this. There's a very, very small proportion of people that live for a long time, and we don't know why that is. And I'd really like to have got hold of their tumours. So the question that I've taken into my research is, why does this fail so badly? What we do here would totally take care of breast cancer. It would take care of your lymphomas and your leukemias, all of those things. What we do would deal with it, but not when it comes to the brain. And so is it something special about the way the brain works, the way the brain is protected? Is it something about those cells themselves? So that's really the problem I've been trying to focus on for the last 10 years almost. Thank you for the reminder. Okay, so we have that blood-brain barrier. This is a problem for the oncologists and the radiation oncologists. The blood-brain barrier stops most drugs getting to them. We have drugs which kill glioblastomas in a dish in the lab beautifully, but we can't get it through the blood-brain barrier. We know that the glioblastoma cells just don't care if you damage them with radiation. They just fix it. And then the other thing we're starting to realise more and more is that the immune system is normally completely excluded. And so where you would normally have an immune response that would help to sort of shut the cancer down, that's completely shut down by these tumours. And so there's nothing that keeps those cancer cells in check. So we've got a really difficult problem. And so this is the problem that occupies much of my waking moments these days, is why don't they die? How are we going to kill these cells? So I'm going to tell you a little bit about our research. Now, I've tried to summarise this, and so I haven't given you a lot of details. I don't have any data pictures for you. I'm very happy to talk about the experiments if you want to, but what I was going to do was just summarise the research that we've been doing, much of which has been supported by the Neurological Foundation. So, before I forget, this is my current research group. I always forget to thank the people that do the work. I sit in my office and I have ideas. I say, go and try this. And one of these brave people will go and try it. And that was at the end of the year. We went axe throwing. Highly recommend axe throwing. Has anyone tried recreational throwing of axes? It's so much fun. Okay, so these are the guys that do the work. So, after lots of thinking, this is essentially the two problems I came up with. And one is, something's fundamentally wrong with these cells. So, they are unkillable. Like, why is that? Can we figure out why we can't kill them? And then the other thought I had is that something is fundamentally wrong with all the research we've done as a community over the last 50 years, because we have made no progress. Now, New Zealand doesn't fund a huge amount of this research, but the rest of the world, everyone in the world is working on this. We've done so much work and we've made no progress. So I wondered if we were doing something really fundamentally wrong with the way that we're doing the research. And that maybe we're using the wrong kind of model system to do that. So, a real quick discussion about our first project. And this is why I told you my long and winding story about how I got into research. Because having worked in all these different areas gives you exposure to lots of different things. And one of the things I noticed when I came into this area was that, in a way, a brain tumour looks a lot like a lymphoma. And I only knew that because I had worked in lymphoma. And what I noticed was that in lymphoma, there's a protein, there's a molecule that lymphoma cells make, and they use it to ignore the fact that their DNA has been damaged and to keep on replicating. And they normally do this because immune cells do complicated things to their DNA, and we don't need to go into that story, but their normal role is to tell a cell, no, you're fine. Your DNA is not damaged. Just keep on doing what you're doing and I'll look after you. Now, that protein, sometimes in a lymphoma, this protein, we call it BCL6, sometimes this protein is active when it shouldn't be. And what it does is it makes a normal cell in your lymph node cancerous, and it makes it resistant to therapy. And I was really struck by the parallel. And I had the realization at the bar at a conference after a couple of beers. And I was so convinced at that stage it was a really good idea. I went back to my hotel room and I started doing some research. And it actually, even after the beers, it turned out to be, it wasn't a total disaster. It actually turned out to be a really interesting finding. So I wondered if the same thing was happening in brain cancer, because the parallels were quite uncanny, and the more I looked into it, the more I could see these unexpected things. But no one who works in lymphoma knows anything about the brain, and no one that works in brains knows anything about lymph nodes, because those two fields in the rest of the world, they would never meet. But in New Zealand, you have to meet people from all these fields, and you have to move around. So because we're so small, I'd worked in so many different areas that I had this insight. And I took a punt on it, and we found that, sure enough, this protein we call BCL6, it is found in brain cancers. It's not found in normal brain cells, but it's in cancers. And the more aggressive the brain cancer, the more of this protein we have. And this protein, we thought was acting as we call it a survival factor. If we block the activity of this BCL6, we can make a GBM cell much more vulnerable. So we think that this BCL6 is part of what makes them unkillable. And the really depressing but interesting part was when a brain cancer cell was treated with radiation, in response, they made more of this survival factor. So it's a really good strategy if you're a cell and you want to live, is that you say, I'm going to make this protein, it's going to keep me alive, I'm not going to die, even though I'm really badly damaged, I'm just going to keep growing. And the more you damage me, the more I'm going to make this protein. So the next experiment we did, well, what happens if we block this protein at the same time that we give radiation or we give temozolomide chemotherapy? Now, in a dish, we've taken someone's tumour, we've mushed it up, we've grown the cells. We can kill these things to like 99% of them are gone by combining this BCL6 inhibitor with the radiation or with the chemotherapy. So we're pretty sure that at least in a plastic dish in the lab, makes them very easy to kill. So we think we've found one of the kind of invulnerability mechanisms. So how about in people? So clinical trials require a bit more data. And so we are working on gathering that data. My colleagues in oncology and in radiation therapy and in neurosurgery, they're interested, but they're quite cautious. They don't want to kill anybody unnecessarily. And this experimental drug that blocks BCL6 is still quite experimental. It's going through the approval process, and it's actually being developed for the lymphomas, but we think we can take this lymphoma drug, and once it's been approved, bring it straight into glioblastoma and start trying it, once we've got just a little bit more data. So this brings me to my second project, which is, what if everyone's just been doing this all wrong for the last 50 years? So in order to get the data that we need to take this clinical trial, we needed to develop a new system. And we wanted a model system that we could work in that wasn't experimenting directly on people, because while there's 100 plus of these cancers a year, there's not enough of them there's not enough people for us to experiment on yet. So we needed to have a model system that we could use in the lab. And we wanted it to look like the human disease. We wanted it to have the same mutations that we find. We wanted it to have the same kind of symptoms that we would see in people. We wanted the tumours to have the same pathology. And we also wanted to see that same suppression of the immune system. Now, that all sounds really sensible. When we look at the work that's been happening over the last 40 years, the models we've been using have not had all of these characteristics. They've had one or two of them, but no one's ever really built a model that has all of them. So that's what we decided to do. We have made a model of glioblastoma that has, all of these characteristics. So this is the work that's been most recently funded, and this was not a sure thing, but what we did was we've genetically engineered some mouse brain cells. Now, I don't work with mice lightly, but this disease is so miserable, and we need to have a system that we work in, that we can manipulate. So we've made GMO mice. We work in a lab, they're all protected, but we have these engineered mice, and they've got known mutations. When we transplant that into our host, into our mouse, we get invasive malignant tumours. They've got the immune system suppression that we're looking for. They've got all the pathology that we want. So we're really happy now that we've got a model where we can start, that's made up of some of the flaws of the previous work, and now we're really starting to get going on trying to understand different components of this. So we're using this for a couple of things. One is to get that data that we want for our clinical trial of our BCL6 inhibitor. The other one is to look at the role of the immune system. So in other kinds of cancers, you can activate someone's immune system. You can give them, they call it checkpoint inhibitors. So Yervoy, I think, is the name for the melanoma. So people who've got melanoma now can get a drug which turns off their immune system. And by turning off the immune system in the cancer, it allows you specifically turn off the interaction between the cancer and the immune system. And what that does is it allows the rest of the immune system to come in and deal with that cancer, to kill the cancer. And this works really well in melanoma and lung cancer and thyroid cancer and some colorectal cancers, but it has failed miserably in glioblastoma. So one of the big projects we're working on now is to try and take these drugs. They're the latest cool things. I went to the Thor movie at the weekend, and Jane Foster, one of the characters in the Thor movie, was being treated with one of these immune checkpoint inhibitors. So they're the latest trendy disease, the latest trendy drug. And so we're really interested in if we can make those drugs work in people with glioblastoma. And we finally now we think we have the right model in order to do that. So hopefully it won't take another 50 years to make some more incremental changes in our research. And so the last thing, I guess, is that the other thing that I'm doing at the moment with the work funded by Neurological Foundation is to try and bring together all the glioblastoma researchers in New Zealand. There's about 4 of us, 5 perhaps groups around the country. And so we're using our model as the centre, like a research hub. And we've got the community together, and we're starting to work together instead of competing with each other. And we're starting to leverage off each other's data and bring our skills together to try and focus our efforts on this disease, because it's been too long that this disease has not been, it's just been too long that we've been able to make just no progress with this. So we're really using this as a hub, a research hub for the whole community in New Zealand to work together. So our system is going to go out to the different groups around the country. They're going to look at it in the areas that they're expert in, and we're going to collaborate as a national community to try and make some progress. So that's a very long talk to get to a very short summary. So I just want to thank all the people that do this. I've been doing this for about 10 years now, and there's been lots of people over the time that have helped me do this. And we've also, so it really does, it takes a village. So I've got some really great collaborators in Dunedin and in the States and here in Wellington. And so I just want to acknowledge that these are the people that do the hard graft in the lab. And these are our funders. So Neurological Foundation have been key in supporting this, and particularly this new project, which has been a bit of a leap for us. And yeah, I'll leave it there. Now, hopefully I've left time to take questions for you. Okay, so people are going to be roaming with microphones. We've got a question here at the back, yeah? Did you have a question?
I do.
Yes, and here's a microphone.
Thank you. If the radiotherapy and chemo doesn't work with glioblastoma, why do you do it? And also, do any of them respond?
Right.
Do any respond to it, though?
So some people do respond.
Right.
So we know that a few % of people will respond.
Right.
We do it because that's what we can do.
You don't know whether they can respond. So
we have no way of knowing if you're going to
be
be someone that responds or someone that doesn't. That's one of the kind of the grails of cancer research is, can we find a way of predicting which therapy will work for you? In brain cancer, we're not there yet. So that is what we can do. We know we can get those drugs into the brain, and we know that in some people they're effective, and in some people they're effective for quite a long time. Right. Mm-hmm. But we have no way of knowing who that's going to be, and that's what we have.
Good. I know someone who's died recently lived very close to a pylon. Is there any research being done on that?
Yeah. So the question there is about pylons. Yes. It's really tricky. The data has not been very reproducible. So there's epidemiological data, like the big population-wide studies say that there's no effect of living near a pylon. There's a lot of anecdotal stories about people. Everyone seems to know someone who lived too close to the pylon. We can't say that it doesn't. When we look at how those kinds of magnetic forces might damage cells, there is a way that this could happen, but we don't know that it does happen. So it's not impossible, but the data has not been conclusive really one way or the other. Yeah.
Thank you for the talk. I always love coming to these talks. My name is Marina, and I have a question about you mentioned about the genetically engineered mice that would then get all the symptoms of glioblastoma. So does that mean that there's maybe some chance that the people that will get malignant glioblastoma would have some specific gene that others don't? Because if you can engineer the mice, then you must know what you're putting in.
Exactly. So we can work backwards, and we can look at people, so we can look at malignant diseases, and we can say, oh, look, these group of people all had a particular mutation. These people have a particular mutation. We don't know what causes those mutations, but we know that if you get a mutation in this gene, in this cell, so in the right cell type, if you get a mutation in the right gene in the right cell, and some other thing happens as well, we don't know what that is, some other thing happens, then you're much more likely to get a cancer. And so this is a really dramatic example for my student who didn't believe this was going to work, I don't think, that we do know, we call them drivers, we know what some of the drivers are. So if we know what the drivers are, we know how to, we should be able to find a way of targeting those drivers. And so that's a big area of research, is finding the mutations that drive the disease and then just trying to get our foot off that accelerator. Yeah.
And just to follow on, is there any chance that the gene therapy could be in the future a solution?
Gene therapy is an interesting idea. I'm not a big fan of gene therapy. I think the trouble with gene therapy is you have to get the gene therapy into all of the genes of all of the cells. And we haven't yet found a way of doing that. Gene therapy works beautifully in kids with immune disorders where we know exactly the one change that's happened in their genes and we know exactly the one cell type. But because in cancers it's much more chaotic, we don't know exactly which mutation, and we know that there's more than one. So I personally don't think gene therapy is ever going to work for cancer. It's just too complicated a disease, really.
One specific type of cancer.
I wouldn't be putting money on it. Yeah.
Hi, thanks for your talk. A sort of a random question. If immunotherapy is going to work as it is random working, along with traditional or consistent medicine so far, or...?
So, back in 2007, we started an immunotherapy trial in GBM here in Wellington, and we tried combining immunotherapy with radiation, but our patient cohort was quite small and we could only follow them for a certain amount of time. Now, last month, a study that started at the same time as us, but they had 40 sites across the US and the UK, and they recruited 500 people, so we could never do something that scale. That has shown for the first time that there is a role for the for manipulating the immune system and getting a positive effect. What they found in that trial, and it took huge numbers, and it took like a 5 to 10 year follow-up, but what they could see in that is that there is a role for immunotherapy in this disease. You have to work out exactly what that role is.
Is that in relation with radiotherapy and or chemo? Or?
Because of the way they designed their trial, they accidentally found a result. And they weren't expecting it, but through an accident of how they designed their trial, they found that it had to be in combination, and you had to get the immunotherapy first and then get the radiation or the chemo. And immunotherapy by itself didn't work, and it didn't work if it came after the treatment. It had to be immune stimulant and then the cancer therapy. So that's the first positive trial in a long time that I've seen in a glioblastoma. And so that gives me confidence that we can get the immune system to work if we just know the right way of doing it. Yeah. And then the government can fund immune checkpoint inhibitors for everyone with a GBM at $100,000 a round, because that's what they really want. The pharmac budget just needs to be a little bit bigger, and then we could fund these things.
What proportion of brain cancers are metastatic? Do you have?
A reasonably high proportion of the cancers that turn up in someone's brain have metastasized from somewhere else. So it's very common for breast cancers to metastasize to the brain. It's quite common for melanoma to end up in the brain. I wouldn't want to put a number on it, but it's...
And is there a time frame? Like, can you say you're safe?
Can you say you're safe after a certain amount of time? It's a bit of a loaded question there. If a metastatic cell is going to turn up, we think they leave those primary tumours quite early. And it depends how long it takes to get to the brain and start growing. But they tend, I'm going to just make some numbers up here. In breast cancer, it's probably in that 5-year sort of range that you'll see an impact. Something like that. Now, there's probably a pathologist out there who's going to completely contradict me on that one, but that's my kind of gut take on that process. We know that those cells leave quite early in the progression of the disease, and it probably takes 5-ish years for them to get established and to start growing. Yeah. I hope that's a short enough window for you. Good. Were there any questions from the live stream? No? Okay, great.
Thank you so much, Melanie, for that fascinating talk. The way you describe the brain and the cancers is just amazing. Even I understand, so thank you for that. And we really wish you all the very best with future research, and what you're doing is so exciting. I would like to say a big thank you to everybody here for coming along this evening, and to everybody who tuned into the live stream as well. Thank you so much. There were a couple of questions on the live stream, but we just don't have time to answer those at the moment, so really, really sorry. And to everybody here, we're going to have a cup of tea or a cup of coffee and some food, so do stay, and if you've got any other questions, we're here to answer them. Thank you so much.